You're listening to Innovating Tomorrow's Medicine with Dr. Jeff Trickett.
In this episode, I'm speaking with Pilgrim Jackson, with over 20 years leadership in pharma and biotech. Prior roles include 13 years at Celgene, where he led multidisciplinary teams in inflammation and oncology drug discovery, headed the system's proteomics group, and contributed to 6 therapeutics reaching clinical trials across multiple modalities. Currently co-founder and CEO of Yatiri Bio. So what treatment modalities do you see as most promising? So whether it's small molecules, biologics, cell therapies, and how does your platform support choosing among them?
If I were starting a drug discovery company from scratch, target through discovery, I actually think it's a good era for small molecules because the biology is more known.
So what makes Yatiri's proteomic platform unique in identifying targets or biomarkers?
One important aspect is that we started with patient samples, not cell lines that so much work has been done on. I would say as far as uniqueness goes, a lot is just that we have built up a really good knowledge base of proteomic data on patient samples. They could not identify sensitive patients. We talked to them, we identified a patient population that was sensitive, relatively uniquely, and clear biomarkers for it. To test it, we actually took from their phase 1 trial, pre-dose peripheral blood samples, completely blinded, and we binned them as sort of stable disease, so a partial response to the drug, holding it steady, and true responders. And they showed us the clinical response, and on the 7 patients that were at that dose level, we were actually 100% accurate, like perfectly accurate.
Pilgrim, welcome. Great to have you here.
Thanks. Yeah, thanks for having me on the show.
So let's dive in with some opening questions. So what inspired the founding of Yatiri Bio, and what problem are you most focused on solving in oncology?
Yeah, I mean, that's a big question for any founder, I would say. And it's been sort of percolating in the back of my head for many years. I was a biophysicist by training, as you know, and went straight from postdoc to Celgene. And in the very first meeting I was in, they talked about using mouse models for predicting efficacy in oncology, but pretty clearly stated was that they didn't correlate with human biology at all. So I raised my hand and said, why aren't we doing something better? And the answer was that this is how everybody does it. And that really just wasn't satisfactory. Yep. So I didn't immediately go out and solve that problem, but over 13 years and working with an amazing team at Celgene, I learned a lot more about the drug discovery and development process and really decided that this was something that we could do a better job of. And then as far as the company itself, it was founded by 4 people, 2 other Celgene colleagues, Mahan Abbasian and Afshin Mahmoodi, and they were actually at Celgene before I joined. So we worked together for 13 years and decided that we liked each other enough to go off and start a crazy startup. And then the 4th member was a really close grad school friend of mine. And so that team really wanted to come out and make a difference, to fill a gap and bridge that translational space and see if we could do something that could actually help patients.
So why did you choose to focus on AML and ovarian cancer as lead indications?
AML was our first indication, and there's 2 reasons. Obviously, fundamental is that there are patients in need. So AML and ovarian cancer, both high unmet need, low 5-year survival rates. There really is a need for new medicines in both of those indications. But in addition to that, AML clinicians are actually very comfortable with precision oncology. So what we were building is these, what we call knowledge maps, so that we can guide therapies to the right patients. That does require that a clinician is comfortable with actually using that guidance to do precision oncology. So AML was first for that reason. They've done it mostly with genomics. Ours has some new layers that are very exciting and much more specific. And on top of that, it is a very heterogeneous disease. So we thought we could do a lot better than the genomic marks that are out there for it right now. Last, but not least, is that it is a very accessible biosample. The tumor itself is in the peripheral blood, so that you can get more dynamic sampling. For our first model, as we were learning how these cancers progress and understanding them, we thought that sampling was of value. Ovarian cancer is our second model. We do want to go into solid tumors. It's clearly a very important area, and also unmet, also heterogeneous. And so we decided that was going to be our second, mostly because of the heterogeneity of the disease.
And I think, as we've discussed in the past, what you're trying to do is sort out the responders from the non-responders. And so I think, AML in particular, such heterogeneity, as you said, so it's a great sort of target to start with.
Yeah, exactly. That's where we get the value. There's also that clear early response in AML that you can predict with a blast count reduction.
So what makes Yatiri's proteomic platform unique in identifying targets or biomarkers?
One important aspect is that we started with patient samples, not cell lines that so much work has been done on. I would say as far as uniqueness goes, a lot is just that we have built up a really good knowledge base of proteomic data on patient samples. As far as proteomics versus some of the other techniques that are out there, it's also unique or important, I would say, because proteins are what drive the biology, of course, and the disconnect between a transcript level and a protein level is actually quite high. So it's very important to incorporate that protein level data. We use cutting edge LC-MS-based proteomics, exploring around 10,000 proteins per sample. So we are at the edge of that, but I wouldn't say the uniqueness is the LC-MS so much as building up a data set for a given indication from patient samples.
How do your proteome models work, and how well do they reflect real tumor biology?
Yeah, I mean, that's a great question, and I'm gonna tie it back to the question you asked about the predictive model as a whole. We talked about the proteomics piece, and that's only one piece of our predictive model. The other piece is functional testing. So our models are built, or what we call knowledge maps, are built with that proteomic data, and then we take patient-derived models, which are the proteome models you're discussing, and we do functional testing on those so that we get perturbations across a large number of biological pathways. And then we bring in the clinical metadata and we mush it all together with some really high-powered AI deep learning. So the proteome model piece that you're calling out here is patient-derived samples, and we've optimized the culturing conditions to really replicate the patient biology. And so we have some kind of unique advantages in this situation because we can test the proteome of the patient, and then we can tweak our tissue culture until those models do maintain a proteome that's very close to the patient they were derived from. So that's what we do. We alter the tissue culture conditions until we find one for each indication that we like. So AML, we're using hematopoietic stem progenitor cells that we maintain in a relatively undifferentiated state. Their proteome does replicate the patient biology quite well. Are they a perfect match? Absolutely not. As things passage over time, they do drift away from the patient they came from. So we do try and maintain them at a very low passage number. And then they also don't capture the full complexity of the microenvironment. Though because we're doing peripheral blood, we do keep some of that microenvironment there. The ovarian cancer ones are completely different. That's a solid tumor. And to do those proteome models, we actually bring in two different aspects. We do bring in organoids that are really the cancer biology itself, and then we bring in microdissected tumor pieces that do try and capture that microenvironment. And again, we can test the proteome as it compares to the parental patient.
So how are your proteome models validated to ensure they reflect patient tumor heterogeneity? And how do you ensure your data set is diverse in terms of patient demographics, tumor subtypes, geographic regions, so that biomarkers or targets are broadly applicable?
Yeah, I mean, that's an excellent question. And I would say there's never enough of that diversity because patients are very heterogeneous. We do it by measuring their genomic status, being sure that we cover, again, I'm going to focus on AML first, we cover the common mutations that are seen in AML patients, and that those mutations are relatively good representation of the patient population. We do it by collaborating with academic medical centers across the country, different regions, as well as international ones in Korea and Japan as well. And then the demographics of age, et cetera, we try and make our models replicate a good sampling of the patient population that's out there. Ovarian cancer, similar answers coming along, a smaller subset that we have at this point. And so we're expanding those, again, with those international collaborations to incorporate more racial diversity.
So what proteomic signatures have you found that differentiate responders from non-responders in AML or ovarian cancer?
We've actually found a lot. So to build the models, we went and did, for AML, we did all the standard of care therapies, all the chemotherapies and venetoclax, combinations, venetoclax plus azacitidine, 7+3, et cetera. But we also put through all the clinical trial compounds that we could get our hands on. Basically, if we could buy them at MedChem Express or SelectChem, we would bring those in and put them through the models. And we've actually found a signature for about a third of the molecules that we test. So it's quite a high number that do have some level of signature, some level of patient selection that would definitely benefit those therapeutics and the patients that are receiving them.
And what role does plasma proteomics play versus tumor proteomics in identifying targets or monitoring treatment?
So we have not done a lot of plasma proteomics previously. We did our work off of peripheral blood mononuclear cells, where you get a much deeper proteome. But the plasma proteomics, it's a very stable biofluid, and we do want to incorporate that. And we did bring in some amazing plasma proteomic workflows with the acquisition of Yatiri Bio AI that just went through a few months ago. So we have some new workflows, and we're incorporating those as we go forward. What we want to do is translate the biomarkers we've discovered to be able to do them in plasma samples as well.
Well, we'll come back to that later, but how does Yatiri integrate biomarker development with therapeutic discovery, and what drives that?
Yeah, that's a great question. So we do it in two ways. We partner with therapeutic companies where we have found a signature for their molecule, or where we think we can find a signature for their molecule. So one great aspect of our knowledge maps is they can be prospective, because the machine learning I mentioned earlier that mushes together that clinical trial data, proteomics data, and functional testing data, that's one purpose, and that sort of puts the patients in these nice pockets of sensitivity. The second purpose of the machine learning is to embed those proteome models into that knowledge map. So now we have something that we can prospectively test empirically. So one way is as a partnership where we help them find the right patients and deliver biomarkers that they can then use in their clinical development and beyond. That's one aspect. What we have realized as we've gone forward is that we actually have a very powerful tool here. And so what we've started to do is identify assets that are post phase 1. So they have a recommended phase 2 dose. They've covered a lot of the normal issues that can hamstring a drug development or drug discovery program. And we take the ones that we've identified biomarkers for, and we in-license those. We try and in-license those and push those forward ourselves. So I would say it's a balance of the two, a platform that's really designed for this repurposing of therapeutics.
And you've done some really cool work with like the FLT3 gene and like collaborations. Can you share some insights into that?
Yeah, absolutely. That's kind of the most exciting recent work. So we were founded in 2020, and a lot of the early stage was a grind building this all up and a dream that we could make those models that predicted clinical outcome better. And recently we've found a number of FLT3 inhibitors that actually have efficacy outside of the FLT3 ITD mutation where they're normally given. So you definitely have a larger percentage of FLT3 ITD patients that do respond, but you have a significant chunk on the order of 10 to 15% of FLT3 wild type patients that can respond to these FLT3 inhibitors and are currently not receiving that therapy. So we think with a biomarker that's driven by our biology, we will increase the response rate, but also include patients that aren't actually getting the treatment right now.
And this could be expanded to other indications outside of AML?
That is a great question that we are trying to address right now. So we are exploring all the options. The obvious spot, not for FLT3 necessarily, but for the expansion of the models is to MDS, and we're definitely exploring that. And it's going to be an interesting learning, I would say. How much can we do with the data that we've derived off of AML? How much can we extrapolate? And how much do we have to build new models for each indication? And so we're exploring that right now with the transition from AML to MDS. And I wouldn't say, as a scientist, I can't give you a solid answer to that yet.
But that's really exciting.
Yeah, it's extremely exciting. And some of the clinical data that we've gotten has been really good just recently.
And I think if you look at some of the drugs that have been more recently like approved in MDS, for example, funnily enough, some of those drugs were actually like discovered, back in the 1990s, and then they've sort of like been brought through sort of like development, and now they're sort of $1 billion drugs. So I think again, it's great for patients.
Agreed. It's a huge area of unmet need with very little to guide the clinicians. And yeah, we're just starting a collaboration with a professor at UCSD to really dive deeply into specifically azacitidine sensitivity and Ven-Aza. I don't know if you know, but the AbbVie just did a huge trial trying to do venetoclax azacitidine in high-risk MDS, the Verona trial. And it failed, but I think that we probably have a biomarker that would separate out patients that are sensitive in that patient population.
Well, I'm sure if you haven't already, you'll be in touch. So
we're working on it. Yes, indeed.
Very nice. So let's move on to targets, modalities, and resistance. So can you walk us through a concrete example where proteomics revealed a novel resistance mechanism?
Well, I wouldn't necessarily call it a resistance mechanism since we're empirical in our work, so much as identification of a population that is sensitive. But yeah, we just 2 months ago got results from a blinded clinical trial. So again, this is a therapeutic from a partner company that had gone through a phase 1 trial. And based on genomic mutations and their understanding of the mechanism, they could not identify sensitive patients. They had a approximately 30% response rate in their recommended phase 2 dose, which wasn't going to beat what's on the market right now. We talked to them. We identified a patient population that was sensitive, relatively uniquely, and clear biomarkers for it. So to test it, we actually took from their phase 1 trial, which nothing had been published on, we took pre-dose peripheral blood samples that they sent us, completely blinded, and we binned them as refractory, sort of stable disease, so a partial response to the drug, holding it steady, and true responders. And then we got the reveal after we emailed back that data to them. They showed us the clinical response, and on the 7 patients that were at that dose level, which is all that we studied, we were actually 100% accurate, like perfectly accurate, which is mind-blowing. So that was the kind of, we made it. We've been working on this. We had the dream for years. This shows that this could revolutionize the way drug discovery is done, really, and drug development.
Yeah, I mean, not to overstate it, but that's kind of the holy grail. If you can say, we basically like mapped out all the different sort of levels of like response. It's 100% accurate. I mean, it doesn't get any better than that.
Can you imagine? Yeah, I will. Funny anecdote on it. The bioinformatician, our head of bioinformatics, was going to present the data to them. We had sent the email. I had seen, an uptick in activity on their side and signing memorandums of understanding and agreement. So I knew it was decent data. But right before the meeting, he's IMing me like all these caveats. Our models are built for this. It might not be this. It could, we don't know, as a bioinformatician does. And then the very first, we started the meeting and they said, let us just show you what the true results are. They flipped that up in the 100% accuracy. So he IMs me. Never mind all that stuff. We're awesome. Yeah, it was a great moment. Yeah, definitely. It was a good time for the company. And it's exciting. If we can extend this to other drugs within AML, other indications, totally revolutionary.
Yeah, for sure. So are there specific resistance pathways or biomarkers that you're particularly excited about advancing?
Yeah, I think so. I would say there's a big range in drug discovery scientists that go all the way from we don't know anything about biology to we can do full rational drug discovery. And I sit on the side that is very close to the we don't know anything about biology. I came out of Celgene. Revlimid and thalidomide were our big drugs that for 60 years nobody knew how they worked, but they worked. So I am more on the empirical and understanding what does work side. Obviously, confidence is increased if your biomarkers for venetoclax work on a BCL2 pathway, and we've seen a number of those. But I would say that what we've found is that at least half the biomarkers we see are probably not immediately tied to well-published biology. And that doesn't reduce their value in my book, because I just think we haven't explored them or we don't understand the connections well enough. So we are much more empirical. So I don't go in with a rational, like, here's the resistance mechanism that we really want to get after. I go after, here's the patient group that is underserved. So our first collaborative project on a target is around the p53 mutant AML, which is definitely the most unmet need. If you have the p53 mutation in AML, it's a crapshoot, and they're not, the outlook isn't great. So we've found targets that are specific in that mutation that we think will have real benefit for those patients.
So what treatment modalities do you see as most promising? So whether it's small molecules, biologics, cell therapies, and how does your platform support choosing among them? Yeah,
so the platform can work really well with small molecules, biologics, bispecifics, ADCs. Theoretically, it should be able to work with cell therapies. We haven't tested it. It does. We do have a number of the immune component cells, the T cells, B cells, NK cells in there. So as far as what do I think is kind of the most valuable, they're all valuable. They all, what does the best for patients is really important. I think it's clearly a hot time right now for ADCs. So you'll see a lot of of those going forward. If I were starting a drug discovery company from where I wanted to do from scratch target through discovery, I actually think it's a good era for small molecules because the biology is more known. I would actually do kinase inhibitor, which a lot of people would be like, oh, that's so boring. That's 20 years ago. You're way out of date. Why aren't you doing something new? To me, now's the time to do that. We understand kinases. We have all these tools that can actually study what's happening in the cell. Yeah, I don't know. I think they all have value. Obviously, for cell therapy, I think the key is to bring the cost down for patients and payers, and then it will have a huge value. At the end of the day, an oral therapy is by far the easiest for patients to work with.
We all like to just take a pill, basically.
Exactly.
So given the aggressive nature of AML, what timelines do you expect from target discovery to IND filing? And what are the biggest bottlenecks? And I know in sort of like chatting about this, I think there's perhaps more of a focus towards having sort of IND ready assets that already been sort of in phase 1 or phase 2. But can you elaborate on that?
I will say on the target discovery, I think our knowledge base, what it can really help with is in identifying novel targets. So more on the competitive landscape side. In that early phase of chemical optimization if you're a small molecule or binder optimization if you're a biologic, we're not going to have a lot of input. We will then be able to accelerate the piece as you're going into clinic, saying that we have high confidence that these will work in the patient samples, in the patients, based on the patient samples, and the target product profile where you have a patient selection hypothesis. We can definitely help with that. So not a lot of acceleration, hopefully higher quality in that phase. And it's that, like, as you mentioned in the question, that's not the piece we really focus on. The part where we can massively accelerate it is the clinical trials. Probably not going to affect your ascending dose phase 1 where it's a tox question. We don't usually address that. But your phase 2 or your phase 1B, we can really reduce those timelines because you can hit statistical significance much more quickly if you're working in a biomarker selected population, so that your response rate is that much higher. Your statistical significance comes much more quickly with a lower number of patients. In addition, clinicians really, really want to know that they're giving a therapy that's going to benefit a patient. So precision medicine has this sort of double-edged sword of, well, if you're limiting patients, do you reduce your accrual rate? I think if you have precision medicine that has some clinical proof of the platform, you will see a massive increase in accrual rate because clinicians will want to put patients on that trial, on that biomarker-driven trial. So even though you're maybe reducing the total population that you're pulling from, that clinician push or pull to get patients onto that therapy with the biomarkers will actually increase your accrual rate and therefore reduce your timelines on clinical trials as well.
Yeah, and coming back to the stratification piece, of course, when you're writing a protocol, you're trying to homogenize your patient population so that you have like nice tight data, and so I think that's how and not just oncology, but all indications. We're going to cure 20% of patients, then we're going to cure 40%, and then 60%, and so on. So I think it's very exciting. How does Yatiri's platform help de-risk clinical trial design in oncology, especially with regard to patient heterogeneity?
Yeah, and I think you touched on it at the very beginning, which is the proteome models recapitulating the patient biology. I mean, I think the biggest risk in clinical trials is in oncology. You make it all the way to your clinical trials after probably a decade of work, right? And significant funds input, and 5% actually make it through the clinical trial process with the biggest chunk falling out in that phase 2 period. So as far as de-risking, it is a matter of just higher confidence that the data you're seeing preclinically is actually going to translate to the patient biology, and that therefore your design, which is based on the patients you're selecting and all of those aspects, now will recapitulate that patient biology, your response rate goes up, and then obviously to me that's the biggest risk of clinical trials, is just not hitting that efficacy endpoint.
So coming back to Yatiri Bio, so with the acquisition, how do diagnostics now fit into your therapeutic pipeline? And where do you seek companion diagnostic status for certain programs?
I mean, it's a really exciting time. We acquired them very end of May. So while it has been a few months, it's, there's a little bit of integration and learning what all is there and how to best utilize it. But really excitingly, it does have a plasma proteomics workflow that's really state of the art. So now we have those capabilities that we can test a very easy biosample. And they have a CLIA, or we now have a CLIA lab, where we can do mass spec-based assays, at least at the LDT level. Right now, the focus is to use that in a companion diagnostic format. As far as early detection, I think it's a vital place for patients and for the population as a whole. Early detection probably would save more lives than anything else. It's an underfunded space. So the plan now is to continue with the Yatiri model, where companion diagnostics is the main focus. But as we start growing, we will explore our own data sets to look for early diagnostics and use our own money to internally try to push that forward as we grow. But no immediate plans for the early diagnostic side of things.
And I think the FDA is increasingly sort of hot or supportive of the companion diagnostics piece. So it's not something you have to have for an approval, but I think you at least have to have shown a plan. It's like, well, this is what we're basically going to do.
Yeah, I do think there was a bit of a turn away and concern around the LDT process, but that has been rolled back, and yes, there's large level of enthusiasm about some level of companion diagnostics, some level of precision medicine. So I do think the overall environment is very supportive of the companion diagnostic piece, and now we can kind of go end to end. Here's the patients that your molecule is going to be efficacious in. Here's the biomarkers to select those patients. And we can help you through a clinical trial with an LDT test that will get you all the way to the market.
How are you thinking about regulatory strategy, especially when it comes to stratified trials or biomarker-linked therapies?
I try not to think about it. No. Yeah, that's an excellent question. We recently brought on a fractional chief medical officer, and so I try not to think about it and make him think about it. But it's really important to us. Right now, we're sticking to an LDT path where we do develop these for the clinical trials and leave it to either our partners for the development of a companion diagnostic, or they can continue with an LDT-based mechanism. I think it's a constantly changing environment, and I wouldn't say I know everything about this. And so what we're going to do in the near future is to start expanding that side of the team and surround ourselves with knowledge that knows more about the regulatory strategy. So
moving on to business and strategic decisions, what is your business model? I'm not asking for the 5-year business plan, by the way. Do you plan to out-license targets, co-develop, or build a full internal pipeline?
Most of the above, but without being distracted. So we sit on an amazing data set of novel targets. We have deep proteome level, deeper than anything that's out there, that's linked to this nice clinical metadata. So you can identify a lot of novel targets. We do not plan on building, skipping to the end, a full internal pipeline starting from novel target. So because of those two pieces, we do want to actually partner out novel targets. And we're starting to work on that off of our data set. Our main focus is going to be the identification, the use of these knowledge maps to identify therapeutics that are deprioritized or shelved that we think have really high value when paired with a biomarker, and push those forward in quick phase 1b, phase 2 clinical trials. So that's kind of the key to our focus, is that ability to identify molecules that have been deprioritized for any of a number of reasons. And it actually means that this horrendous environment that we've lived through for the last 4 years for startup biotech, for biotech as a whole, really, is potentially to our advantage, because now we have this tool that can actually prospectively look and empirically look at molecules and identify ones we really want to pull in. And so since companies have had to streamline and focus on a single asset, there's a number of assets that are sitting just on the periphery that are very good, that did get deprioritized for financial reasons and non-scientific, and we can start to scoop those up.
So what is your approach to partnerships with pharma, and what makes a successful collaboration in your view?
Yeah, and I didn't, I guess, answer your last question that thoroughly. So we partner out the novel targets, but we also want to use our knowledge maps to partner positioning of therapeutics for a pharma where it is their front runner. Our main focus is definitely being able to use them on our own assets. But to me, I mean, the founding team built the company because there's a gap, and we want to always use those knowledge maps and the knowledge we've built to really help patients. So our approach is to partner as often as possible where there is benefit. The partnerships where we do guide patients and identify patient subsets is tech access plus milestone based partnership, where we are in the game with the company, where we're betting on ourselves, and that we do put clinical milestones as they progress using our biomarkers. And then what makes a good partner? The more open and scientific the discussion can be, the faster you can move through and really address critical issues. So I would say openness, clean communication, those are good. Definitely not really, really long, drawn out MSA back and forth. That's not good. Clean and quick and getting the business alignment quickly seems to be what we've found as good partners.
I would agree. I think I won't say who, but I think my record for an MSA was actually 2 years to renegotiate it. So it took a while.
I hope I don't beat that record, but we've gotten a few that are getting close. Yeah.
I hope you don't either. So how do you prioritize new indications beyond AML and ovarian, and what criteria drive those choices?
Yeah, I mean, that's an excellent question that we're kind of grappling with all the time, and especially right now as we bring in our first asset of our own. I would say the major criteria that drives those choices is financing and the ability to focus the team. Like, how many team members do we have that can think deeply about a given project? To me, one of the things that we do really well is that we go to the depth that's needed on the biology. We're not throwing AI at something at a superficial level and saying, look, we've solved it. We're really deep on the biology of each indication. So I think a lot will depend on the financing. I wouldn't say it's a finalized decision. We definitely want to do our AML and the expansion into MDS, so the heme arm. We do want to see that it crosses over to the solid tumors. And behind that, we have built up collaborations with academic medical centers for additional solid tumor indications. Right now, it's basically prioritized on financing. So if my ideal would be we build some of those in partnership with the pharma or large biotech that actually wants to have early access to one of those models or exclusive access for a period of time. We build it together, we get the model, they get the knowledge that comes out of it for their molecule.
I think we're at an interesting time in terms of like the financing. It's not been great in sort of like in recent years, but I think just having spoken to some like biotech analysts like recently, and actually just taking a step back, a sort of general question is like, who on earth would invest in biotech? You may as well go to the casino in Las Vegas. You actually get better odds putting your money on black or red. So who on earth? But with that said, I think we're at an interesting point in history. So like, if you look at the XBI index, which I think started in 2006. So it's almost 20 years since it's been going. And of course it includes, I think it's about 120 different companies actually.
It's been 20 years since 2006.
Yeah, right. Yeah. But it includes companies like Amgen and others. But what's happened is that for the first time ever, this index is actually showing profitability. And so of course investors are taking notice of that. And I think what has changed is that biotechs, people in general have actually worked out how to do successful commercial launches for products. And part of that comes back to sort of like appropriate reimbursement, appropriate sort of like pricing. And so some of and you mentioned like small molecules. So because of the cost of goods, is relatively small compared to cell and gene therapy and other ones. Yeah. They're actually very profitable. And so because of this combination of factors, I think we're starting to be in a different place. I'm not saying we're out of the woods yet, but I think there is reasons for optimism, and you're already seeing some large acquisitions, even here, especially here actually in San Diego. I think AbbVie, you mentioned AbbVie actually bought like Capstan Therapeutics for I think a couple of billion. So the things are starting to change. So I think it's not without its challenges, but just sharing that sort of some words of optimism, I suppose.
I have the same feeling, Jeff. I would agree. Little shoots of green coming through the frozen tundra, and it is good. We're gearing up for a Series A launch here soon, and I have a good feeling that the environment has shifted from the last couple years.
One of my favorite questions, actually. So what does success look like for Yatiri in, say, the next 3 to 5 years?
Yeah, I mean, that's always a fun one to think about, of course. I want to build the knowledge maps, and it does tie back to how many, like getting to different indications. Ideally, we would build these knowledge maps for a number of the key oncology indications. So 3 to 5 years, mostly oncology. I do think that our ideas will translate to I&I and neuro. And so beyond 5 years, we would want to start exploring that as well. But I would love it for Yatiri to be sort of this central hub that is really a data company, that is a knowledge company, I should say. And so we have knowledge maps for all of these indications, and that we can start treating oncology as a collection of rare diseases, which is the way I view the cancer as a whole. So we can start treating it in that manner, and that out of that, we can have spin-outs of molecules where we've identified a particular molecule, a particular patient subset that has high unmet need, and we have a hub and spoke model where around the Yatiri Bio knowledge are a number of companies that have molecules that they're pushing forward for given patient subsets.
I do like the word knowledge map. I think I've used the term knowledge asset. I mean, it's kind of the same thing, but I think a lot of these diseases are actually still very poorly understood. And so I think, being at the center of that is, that's a great place to be.
I completely agree, especially with all the tools that are becoming available to sort of utilize this disparate pieces of knowledge, and being able to integrate that and actually create something of value is the future.
What advice would you give to first-time biotech founders navigating complex translational science?
First to first-time biotech founders, I'd follow in Jensen Huang's recommendation and say, don't do it. And then after that, it would be, I think you have to be very passionate about what you're doing and follow the science. Like, dig deep. Don't get distracted by superficial wins of venture capital, where what's really popular right now or what pharma's the most interested in, because there is a herd mentality both in pharma and venture, and it's natural. I'm not trying to denigrate any of those guys since I'm going to go ask money from both of them, but there is some level of herd mentality, and I think if you want to do something truly significant in translational science, you're going to have to identify that, be passionate about it, and sort of stick to the scientific aspect that you're discovering and the data that you're uncovering.
So as we wrap up, are there any questions that I didn't ask, or are there any final comments that you wanted to share?
No, I think you've covered a lot of it. I don't think we maybe talked as much about sort of that balance, because we have the partnership model of revenue and the therapeutic, which is the home run shot, and negotiating that with venture. So I would say a question sort of around that. It's been one of the more complex pieces for me to handle as the CEO and the one out begging for money. Is that balance. And early on, it was very much forget the revenue side, focus on the therapeutics. But being honest, if we had focused on the therapeutics, we probably wouldn't have been able to survive that extension of runway. So I do think there's going to need to be a transition in the funding side of companies where there is some balance of real company generation and revenue and the moonshot therapeutic side of things. They are valued completely different. I do understand that it can be a distraction, and building a company and the partnership piece can be complicated, but I think it does strengthen the company and the team to sort of understand that value.
It's a good point, because of course the question you're going to get is, well, it's like, are you a platform company? Are you a therapeutic company? And different people sort of say, well, you can't be all things. You have to pick one. But I don't see why you can't actually have the best of both. And I think, as with all things, there's advantages and disadvantages. So being a company that is revenue producing, well, that's just a good place to be for lots of obvious reasons. And I think that actually makes you more attractive to potential sort of investors. So I think I think, and especially like on the knowledge map side and the data sort of like side of thing, again, I think that's how we're going to help. And you mentioned, other therapeutic areas outside of oncology, so neuroscience and I, and again, those are, if you look at the R&D spend, those, not surprising, those are sort of increasingly sort of massive areas for R&D spending. So I think those are a good place to be. But I think I would just encourage you to sort of keep doing what you're doing. Thank you, and a great conversation.
Thank you. Yeah, thanks, Jeff.
Innovating Tomorrow's Medicine with Dr. Jeff Trickett.